So we're at the top of the hour. Very excited today to have Dr. Joanne Boldison. She's currently at the University of Exeter Medical School in England. Oops. And
So,
sorry, the title of her talk is Dissecting the Role of B Cells in the Pathogenesis of Type 1 Diabetes. Just a little bit about her. She completed her degree in applied biochemistry and molecular biology in 2006 at the University West of England, which included a year as a visiting undergraduate scientist at the cell biology department at the University of Cincinnati in the US. Following her undergraduate degree, she took up the role of a laboratory technician at diabetes metabolic unit at the University of Bristol, and then she undertook her doctoral studies in the Faculty of Medicine at University of Bristol with a focus on autoimmunity. In particular, her research into ocular inflammation that focused on CD8 T cells and their regulatory mechanisms in a model of experimental autoimmune uveitis. And following her PhD, she moved to Cardiff University to continue postdoctoral studies with Professor Susan Wong, studying the immune pathology of type 1 diabetes. So as I mentioned, she's currently at the University of Exeter Medical School, and her research interest is to increase understanding in the immune pathology of type 1 diabetes, focusing specifically on the role of B cells in the inflammatory process and how they interact with other immune cell subsets. Specifically, number one, dissecting the B cell dendritic cell crosstalk and mechanisms during the development of disease and uncovering immune dysregulation in individuals diagnosed with type 1 diabetes. Number two, identifying the role of islet-specific B cells and their importance in pancreatic beta cell destruction. And three, understanding B cell cooperation with immune cells and pancreatic beta cells during disease development. I'm going to drop her recent paper, Gene Expression Profiling in NOD Mice Reveals that B cells are highly educated by the pancreatic environment during autoimmune diabetes. I'll drop that into the chat. And thank you very much for joining us. Welcome.
thank you for having me. Thank you for inviting me. I feel very grateful to come and share some work today. Thank you for the lovely introduction. It was like, this is your life.
Yeah.
Can everybody hear me?
That's great. Well, that's where we provide it. And I think it kind of illustrates a really interesting trajectory to where you are now.
Yeah, I've taken a strange path.
No, I think that I think actually sort of like this interdisciplinary route really fosters new ways of looking at this 100-year-old plus disease.
Yes. So if everybody can hear me, I shall jump straight in.
Right.
And yeah, so just reiterate my thanks in inviting me to talk about B cells today. And so I will dive right in and talk about B cell involvement in type 1 diabetes. And so this is a schematic here that I think just sort of highlights some of the key mechanisms. And I think everybody knows how important B cells are in terms of their antigen presentation and their ability to present islet antigens to both CD4 T cells and cross-present antigen to CD8 T cells. And this encourages islet immune attack and the destruction of beta cell insulin-producing beta cells. And as a consequence of this immune interaction, these B cells produce autoantibodies, islet autoantibodies that everybody knows is used as a clinical marker. But I think one of the things that's been really unclear and we really don't know is how these B cells are behaving in pancreatic tissue during diabetes development and disease. And so here I've just given you sort of a few mechanisms of B cells that could be doing some key damage here in the pancreas, where we've got islet reactive B cells presenting antigens to T cells and being activated and releasing cytokines and encouraging that CD8 T cell cytotoxic killing to destroy beta cells. And then we have the possibility that these islet reactive B cells are not presenting antigen, but maybe being activated by certain cytokines, such as type 1 interferon, or receiving signals from through their TLRs, so their toll-like receptor, and producing cytokines. Or it could be that we've got a population of non-specific B cells in the pancreas that are undergoing a similar mechanism and contributing to that pro-inflammatory environment with cytokine release after being activated by a similar mechanism. And so really what I've been trying to focus on and answer in the last few years is understand if, number one, what these B cells express. Are they of the same subset? Are they of different subsets? And how they function in the tissue. And so this is what I have been trying to focus on, enable to us to be able to understand what they express, and then be able to see if we can pull them out in peripheral blood of donors for us to be able to study them and target them. But I can't really talk about B cells in type 1 diabetes without talking about the work from my colleagues here at Exeter, so Pierre Leet, Sarah Richardson, Noel Morgan, and their sort of seminal studies in the pancreas looking at B cells using immunofluorescence and showing that CD20 positive B cells are indeed expressed in the pancreas, but there are the two subsets of donors. One subset of donors have a high number of CD8 T cells, and CD20 positive B cells were associated with high levels of inflammation, increased loss of beta cell mass at diagnosis, intended to be diagnosed younger. And then on the other side of the coin, you have individuals with lower numbers of CD20 positive B cells, a lower grade of inflammation, a higher beta cell mass left at diagnosis, and tended to be diagnosed older. And I think everybody is aware of this work now, and these have been coined either CD20 high donors, CD20 low donors, type 1 or type 2, or endotype 1 or endotype 2, depending on what you want to call them. But this has really been quite important about how we think about B cell research in type 1 diabetes. And so really, I think one of the questions that we all have are, can we improve targeted B cell therapy in type 1 diabetes? I think everybody is aware of the rituximab trial and the delay in the loss of C-peptide, and the numerous preclinical studies that have undergone in the NOD mouse model targeting B cells, whether that's targeting CD20 on the B cell to deplete them, targeting different receptors on the B cell, or targeting the survival cytokines of these B cells. So there's been numerous ways of being able to target B cells, and which have showed a delay in the onset of diabetes and even reverse diabetes onset. And so these are some of the studies that I did when I was with Susan Wong at Cardiff, and these had a sort of highlight of trying to understand what happens when we remove B cells to that pancreatic environment, and what happens to the pancreatic environment when those B cells return. And these have been published for quite a while now, so I'm not going to talk about them today, but it's sort of the journey, the B cell journey that I've been on in the past few years. And that work has led to this specific work that was published last year, and which I've condensed into one slide, so a whole paper into sort of one slide to sort of lead you onto some of the unpublished data that I wanted to show you today, that I think is really exciting. But I sort of wanted to set the scene of where that's come from, and sort of the questions that I'm trying to answer. And so some of the work that I did towards the end of my postdoc with Susan was done in these NOD mice. And so if you're not familiar with the NOD mice, they're a non-obese diabetic mouse model. These mice develop spontaneous diabetes on account of their genetic susceptibility. And in this particular study, I'm taking B cells from the pancreatic lymph node and the pancreatic islets from animals that are about week 19 to week 20. And so that means that, and they weren't diabetic, but they were at a stage of diabetes onset. So there's the balance where you have lots of immune cells to study, but we can still isolate the pancreatic islets from the pancreas in order to be able to actually isolate them properly. And so one of the key things throughout this paper was the gene expression studies that I'd done. But one of the things that were really highlighted was the difference in the B cell populations that were in the pancreatic lymph node in comparison to the pancreatic islets. And you can see this on the principal component plot here after I've plotted all the genes. And this was really highlighted when I looked at the genes that were expressed, and it was a number of interferon stimulated genes. And there's a whole list in the paper if you want to go and have a look. But what I really wanted to highlight here was the upregulation of TLR7, and I confirmed this using immunofluorescence. So this is a slice of pancreas where I've stained for CD20 and TLR7 in the green, CD20 in the red, and you can see these double positive B cells. And so this was sort of what led me on to the next bit of work, and trying to understand or ask the question, do these particular B cells that we see in the pancreas, do they have a propensity to upregulate TLR7 once they're in that environment? And so one of the things that I really wanted to do when I got my fellowship at Exeter was trying to translate my work from mouse into man, and to be able to have both strings to the bow, I guess, and be able to dissect how B cells are involved in diabetes in both the mechanisms in mouse, and being able to translate those observations. And so one of the things that were particularly piqued my interest in the literature after getting the TLR7 data back was the fact that these particular B cells had been noted in other autoimmune settings, mainly SLE, and now much more is coming out, such as more research in infection and other autoimmune diseases. But these particular B cells had TLR7 sensitivity, and they're termed DN2 B cells, but they can be found in this particular double negative subset of B cells in the PBMCs from peripheral blood in healthy donors. And so you can see that I've marked CD27, the traditional gating of CD27 and IgD. So you have your switched memory, your naive, your unswitched memory, and this double negative compartment. And this paper came out from Ignacio Sanz and colleagues, and this is their abstract diagram, which I think is a really nice diagram of explanation of what they found. And so what they found was that these particular DN2 B cells were TLR7 sensitive. They express CD11C, and they're precursors to antibody secreting cells. And so before I sort of move on to some of the data that I've got, I sort of wanted to set the scene and make sure everybody was up to date on the DMB cell pathway. And because some of the data, some of the literature out there is relatively new, some of the subsets haven't quite been, fully explored yet, and there's still quite a lot of unanswered questions. So this might look intimidating, but I'll walk you through it. And so traditionally, I think everybody is aware that you have your naive B cells. They can enter the germinal center, undergo clonal expansion, class switching, and differentiate into your class switch memory population, your CD27 IgD negative B cells in the traditional sense. But now there's been identification of a DN1 subset, so they're double negative, so CD27 IgD negative, don't express CD11C, but remain CD21 positive and CXCR5 positive, and they possibility as a precursor to these traditional class switched memory B cells of what we see in a traditional sense. And when I say that, I mean, sort of the classical CD27 positive, IgD negative B cells. There are DN4 B cells, but they're very, very few in the peripheral blood, and haven't really been much explored yet. I think there's maybe one paper with the fact that they might have something to do with IgE class switching. So I'm not really going to talk about them today. But the two sets of B cells that were sort of piqued my interest really are these CD11C positive B cells that have this TLR7 hypersensitivity. And so this is an alternate development pathway outside of the germinal center, so it may be extrafollicular associated. And so you can, from the Ignacio Sanz paper, they found that activated naive B cells may be precursors to these DN2 B cells, both sharing CD11C expression, but either in the naive compartment or the DN compartment. And then there's been further work on the DN3 subset, which don't actually express very much at all. They lack CD11C, lack CXCR5, lack CD21, lack CD27, and lack IgD. And so they don't really seem to express very much in terms of the B cell markers at all. But they have also been suggested as a precursor to these DN2 B cells. And so one of the first things I did was to ask the question of are these DNB cells enriched in type 1 diabetes? And so first of all, I looked at the whole double negative B cell subset. And so I took donors with a type 1 diabetes diagnosis, whether they were newly diagnosed or whether they'd had diabetes for a longer duration, so a long standing. And then I did some flow cytometry on their PBMCs and did some traditional gating of the CD27 and the IgD to mark the DN B cells and all these four subsets. And I've just popped the T-SNE plots on there so you can actually see the fact that the DN B cell do cluster on the T-SNE maps nicely. And when I collated all the data together, I found that there was an increase in these DN B cells in long-standing donors, and I didn't see much of an increase in the newly diagnosed donors, and I saw a reciprocal decrease, a contraction in these unswitched memory population. And so following this, I made sure I was actually looking at the CD11C population. And so looking at the CD11C population in this subset here and in these naive populations, so the activated naive population here, I didn't see any significant increase in the CD11C positive cells that are in the naive subset, but I did see an increase in the CD11C positive cells that were in the double negative subset in the long standings. And so instead of, I sort of went one step further with the analysis really because of the fact that thinking about the DN2 and the DN3 and the DN1 maybe from a different pathway. And so I gated DN1 cells, DN2 cells and DN3 cells on account of their CXCR5 and CD11C expression, and then took the CXCR5 the CXCR5 negative population and looked at the CD11C, and you can sort of clearly see the difference in the population there. And again, when I collated the data, I found an increased loss in CXCR5 cells in the long standings and some in the newly diagnosed, an increased frequency of the DN1 cells as a percentage total of the CD19, and an increase in the DN2 and the DN3 cells as well, as a percentage frequency of the total B cell population that I downsampled in the flow cytometry data. But when I looked at the proportion, you could clearly see the switch from the loss of DN1 into more of a DN3, DN2 phenotype in the donors with type 1 diabetes. You can see that this population, the DN1 is sort of contracted, and the DN2 and the DN3 is increased. And so I just wanted to note that the DNB cells in terms of their phenotype, so there's been quite a lot of that they express TBET and CD11C, but actually I haven't been able to find much data in the fact if they express TLR7 on their surface, only gene expression studies. And so I wanted to confirm that these particular DN2B cells do have an increase of TLR7 in terms of their protein expression. And I confirmed this looking at the DN1 subset, DN2 subset, and the DN3 subset. And you can see the difference in the shift of the TBET. They have an increased CD19, this DN2 population, and they lack CD38 and CD24. And when I looked at all the healthy donors together, you could really see that I was pulling out the DN2 B cells that expressed a high level of TBET and a high level of TLR7, which may account for their hyperresponsiveness. And so I think one of the clear questions that would follow is there a difference in phenotype in type 1 diabetes in these particular B cells? And yes, there is. I found quite a significant loss of Tbet in the DN2 subset in type 1 diabetes donors, and but no difference in the TLR7 expression. I found a slight reduction in CXCR3 across the board in these particular donor subsets. The DN2 reached the lowest significance. And I also found a significant increase in CD95 in these particular DN2 B cells. So certainly this might indicate that they're functioning differently because they have a higher CD95, which is B cell activation, or it might be that their loss of Tbet is maybe some internal regulation of their class switching abilities, as that's what Tbet is known for in B cells. And so that's sort of a undefined question that I'd like to answer going forward. But certainly it's these DN2 B cells that seem to have an altered phenotype in type 1 diabetes. And so one of the interesting things is when I noted looking at the data is that the DN2 and the DN3 and sometimes the DN1 is that some donors were quite variable. Some donors could be quite high for DN2, or some donors could be quite high for DN3. And so I wanted to sort of understand this a bit more and ask if these donors were these donors and whether DN2 was associated with DN3, so I could, sort of see if it was if more DN cells meant more of everything. And so I did some clustering analysis of the DN subsets in each of my donors. So each of the mark on the grid on the heat map is a donor, so it's a mix of the newly diagnosed and the long-standings. And when I clustered them, it looked really clear that you either have DN1, DN2, or a DN3 phenotype. It wasn't that if you had more DN2, you had more DN3, you had more DN1. It looked like that there was actually like distinct DN subset donors. And so here you can see that you've got a bunch of donors here that cluster into the DN1 donor phenotype, where you've got a lot of DN1 and not much of the DN2 and the DN3. Whereas at the top, you seem to have a reduction in this DN1, not much DN2, but quite a high expression of these DN3 cells. And then on the bottom, you have these DN2 phenotypes where you have less DN1, but more DN2 and not a lot of DN3. And so this to me was really quite interesting that they seem to cluster out, and that it wasn't just that you have increased frequency of DN double negative B cells, but you actually have an increased frequency, or you have a specific phenotype in terms of which DN cell you have that's enriched. And so when I took the data and had a look at the DN frequency, the DN percentage in the frequency of CD19, you could see that it's these DN2 and these DN3 B cells that seem to have the highest frequency of the whole double negative B cell subset. There were some donors that were DN1 in the DN1 group that had a higher frequency of these double negative B cells, but it seemed to be the majority of them were in these DN2, DN3 groups. And the same with the contraction of that unswitched memory compartment. It seems that there was more of that switch into in the DN2 and the DN3 groups. And so one of the things that was sort of a question to me is why have these donors got, what does it mean? Are these all males, or all these females? Am I picking out donors that have been diagnosed at a different age, or if they've had a longer duration? And so what I did was took the clustering map and put lots of donor demographics against it and tried to understand if there was some sort of association with different donor demographics, or sort of who I'd collected in my study. And you could see that I looked at sex, age of diagnosis, and duration. And so in the DN2 and the DN3 subsets, it seemed that there was more females in the group, but it certainly wasn't exclusive. There were males in the group of the DN3 and the DN2. Age had no correlation when I actually did the correlation. It was sort of a mixture of people. The age of diagnosis, in the white bars, I should say, are the newly diagnosed, because they're all diagnosed as adults. We don't have any children coming into the ISTID trials at Cardiff, and so these are the newly diagnosed are all adults, and so I've just left them blank because they've sort of only been newly diagnosed as adults at 6 months. But you can see that in terms of the long standings, that the DN2 subset B cells didn't have any young diagnoses in. They were all sort of older, whether they were diagnosed at teens or adults. And then there was a sort of a few dotted around that were diagnosed younger in the DN3 subset. But certainly, again, not exclusive. There was, donors that were adults and diagnosed and had as adults in the group. And so there was no correlation. And the same with duration. It didn't seem that there was any correlation in terms of the of how long they'd had diabetes for.
Joanne, can I just ask a quick question about that
Mhm.
slide? If
Want to go back?
you
I'll
can
go back.
expand, oh yeah, thank you. If you can expand the sample size there, do you think you could get more clarity in your signal?
Yeah, definitely.
And like how, it looks, how many people were involved total, and how might you, is there a repository where you could get more of these samples, or this is something where you have to like start from in the clinic?
Yeah, so I had 23 long standings and 14 newly diagnosed.
Mhm.
Uh, and so quite a small donor subset. I think if we did expand, you might see some stronger correlations. And it also might be that there might be like a mix of correlations in that, sort of, and you sort of layer up those factors that make you a DN2 phenotype or a DN3 phenotype, and we might be able to do that if we did have more donors. In terms of where I can get the donors from, I'm applying to Address 2 to see if I can get PBMCs and be able to study the B cells for a larger number of donors that I can access, and certainly they would include donors that have been diagnosed young as well. And so that's part of the future plan, is to hopefully be able to access more donors, to be able to add into this these data sets to see if we can be able to pull out any of those correlations in that are sort of gives us maybe a hint now, but it might be sort of stronger as we get more end numbers. And I've plans to do some single cell sequencing on those particular PBMCs and stuff. So yeah, does that answer your question?
Yeah, I think Rachel Bonamy is at Vanderbilt, and she's doing a lot of work on the B lymphocytes as well. Who knows? Maybe they've got a repository that you guys could cross collaborate with.
Yeah, so we still get donors into Cardiff. They tend to be sort of filtered off into different sort of trials that are running, and we get people who are either not suitable for the trials, or they'd like to give blood and be a part of the research. And so that does continue. I can still collect, but it's not a huge amount of, it's not hundreds
Right.
of donors. And so yeah, hopefully that we'll be able to get access some samples from Address 2, and and so anywhere else that would be willing to
Well,
sort of donate.
it's a great first look and foray into these
Yeah.
data. And it's really interesting what you've already kind of been able to pull out. So it's exciting in terms of where it could go next, It's really great.
Yes. And so if okay, I'll continue.
Yes.
And sort of add a couple more slides of just about the sort of those associations with the DN subsets of donors. We did look at genetic risk and looked at HLA genetic risk score after doing some typing for the DR4 DR3, etc. haplotypes, and used this that's been previously published, and looked at the genetic risk score of our DN1, DN2, and DN3. And again, I didn't really see any striking differences. It's not like there was one particular subset that had a lot more, you know, at a higher genetic risk score. And you know, maybe that it might be different if I had a large number of donors, but certainly nothing jumped out at me in terms of there was quite a variation in terms of their genetic risk score in each group. And we did also look at the SNP associated with PTPN22, so the SNP associated with type 1 diabetes, and we found a handful of donors that had that particular T switch from the C, but when we looked at those DNB cells as a percentage of CD19 with each of the PTPN22 types, we didn't really find any difference between the each group in the type 1 diabetes group. And so it didn't seem that, you know, all the DN2s were you know, had the different allele. And so one of the things that did come out, though, which I thought was really interesting, was that when I correlated the C peptide with the percentage of DN1, I didn't find any significance. But when I correlated the C peptide with DN2, I did find a significance in a negative correlation. And what was even more sort of apparent was when I did the DN2 to DN1 ratio of these in these particular proportions of cells, you could really see that the ones that had the higher DN2, DN1 ratio, the C peptide was on the baseline, and you can almost draw a line here where you can see that those particular donors of that, you know, DN2B cells correlate with the lower C peptide. And so I found this quite interesting. And during sort of the time I was collating the data, I was already asking the question about, you know, sort of what, you know, those B cells are in the human pancreas. And so are these DN2B cells present in the pancreas of these type 1 donors where they have a high CD20 phenotype and more high grade of inflammation? And so with the help of Katie Morrill in Sarah Richardson's lab and my postdoc Sophie, we had a look and see if we could detect CD11C B cells in the using the 4 donors in the EADB biobank. And so these are recent onset donors that have been marked as a CD20 high. So we chose donors that we know that had CD20 positive cells for us to be able to detect them. And I'm just showing you some images here, but I wanted to draw your attention to the orange arrows where you can see the CD20 positive B cells that are double positive for CD11C. You can see here this orange arrow is the cell that's positive in the red, and you can see the CD11C staining in the green here, and there's a few of them dotted around. We've also been able to detect CD27 positive B cells that aren't positive for CD11C in the pink, and you can see the one example there, but you know there's more pictures. And so when we did the quantitative analysis for this, and this has just been done, so very new data, we looked at immune regions of interest and insulin containing islets. You can see here there's a graph of each donor and their percentage of insulin containing islets. So I'm just wanting to note that actually one donor didn't have any insulin containing islets left. And then this is the frequency of insulin containing islets. And we chose that to look at immune regions and insulin containing islets that had over 5 CD20 positive cells, so we could look at those particular regions that were heavy in CD20 positivity. And so you can see the frequency of the CD20 positive cells here. And so we've got the different phenotypes of CD20 in the immune regions, in the crosses and in the circles. the insulin-containing islets that were remaining. And you can see that the double positive B cells are certainly there, but they are in very, very few frequency. And so when we actually look at the proportion of B cells, they make up both in the insulin-containing islets and in the immune regions only about 1%. There's a reasonable slice of CD27 memory B cells. They could possibly be unswitched memory B cells, but they certainly express CD27 along with the CD20. But there is quite a large proportion of B cells that do not express CD11C or CD27. And so a lot of this data has sort of given me a lot of questions, more questions than answers. And so, but I think one of the things that we can confirm is that certainly the CD11C positive B cells are there, certainly not in high frequency in the 4 donors that we've looked at. And this is something that I think that definitely needs to be explored more. And it might be that these cells are DN3, so whether they're not expressing CD27, not expressing a naive marker, and not expressing CD11C. And so there's work to be done, but I think it's exciting, and it leaves me with lots of unanswered questions as usual, and as science should, I think. So some of my unanswered questions are: what are the phenotype of those remaining CD20 B cells that are unidentified in those Tide 1 donors? What is driving the increase in DN2 B cells in that subset of individuals that correlate with the lower C peptide? And do we see an increase in DN2 B cells in younger onset type 1 diabetes when the individuals in children? Is there a DN2, DN3 phenotype that's more associated with being diagnosed young? And lots of other questions about the functionality of these cells that need to be answered. And so I'll leave it on my unanswered questions, and I'm happy to, if anybody wants to answer them for me, then wonderful. But I'll just sort of say my thanks to everybody, especially people at Cardiff and the ISTID team, where I've been able to collect the samples. Susan and James have been brilliant in allowing me to sort of be able to work at both Exeter and Cardiff, for me to be able to pull together as much of the resources as possible. And of course, Noel and Sarah at Exeter, and Katie for helping with the staining, Pia, everybody in the IBEX team, and my new postdoc, Sophie, who did some of the analysis, and of course, all the donors from the biobank, and my funders, past and present, for believing in B cells and research in type 1. So I'll take any questions. Thank you.
That's fantastic. Really interesting work. We'll open it up to questions. If anyone has one, please drop it in the chat, and or you can raise your hand, we'll unmute you. Yeah, it seems you know, that there's this is sort of like the tip of the iceberg, right? So you've got to go now and really replicate with many more samples. Oh, here's a question from Sarah Richardson. Another
I
rock
thought
star.
Sarah might have a question.
Yes, beautiful talk, Joe. Well done. I think the address 2 work would be very informative, as most of your young donors do or have a longer duration of disease. Interesting that your long-standing donors, which include those younger donors, are the ones that you see the most differences in. Do you want to comment on that?
Let me get might help if I get the chat up, then I can actually see.
It does help.
Yeah, it does, yeah. So interesting that your long-standing donors, which include the younger donors, are the ones you see the most differences in. Yes, so I do see the most differences in the long-standing, but they do have a variable duration. And so, I mean, I can do some further analysis, but when I did correlate the duration with any of those DN2, DNC, DN3, the duration of the disease didn't make any difference. But yes, I do tend to see more of the CD11Cs in the long-standings, but I had more long-standing donors. So I don't know if I'd be able to collect 10 more newly diagnosed, that may balance out, you know, and we'd actually see, you know, sort of, I guess when I analyze them. I don't know whether they're going to be have a DN high phenotype or not, or have that enrichment of DN2.
Um, let's see. Okay, that's great. One from Josie Clarkson, really fantastic talk, Joanne. It's my first time hearing you speak. It describes such complicated, to me anyways, processes so eloquently and clearly. I'm excited to see how you approach the unanswered questions and get some more answers that could lead to some promising cell target candidates. So it's some very great, really nice accolades in the chat. I wondered, we did create together with our partner, Sugar Science, together with our partners at Felix Braden and company at Air Data Commons and the iReceptor group, a T1D TCR repository. We started that out, and Stephanie Hanna is part of that group from Cardiff. And
Yeah,
then now we've got people coming in with B cell receptor data as well, genetic
yeah,
data. And so, Rachel Bonamy, as I mentioned, she's a big beta cell person at Vanderbilt. And so she's part of the effort as well. So it might be something to think about if you guys are interested, bringing in some of these data once it's sorted to kind of develop some kind of a landscape
yeah.
of what the B cells are doing, because it's really looking like they're quite involved and important.
Yeah, I've
And
spoken to Steph sort of, about, sort of the B cell stuff, the B cell receptor sequencing that she's been doing,
Mhm.
and the plan to do some, which is now funded by Helmsley, is to do some single cell sequencing and B cell receptor sequencing. And I've been taking lots of advice from Steph about that, and so hopefully we'll be able to do that single cell and the B cell receptor sequencing on the address 2 donors, where we both have adults and younger onset donors, to be able to really pull out on a larger subset. What if I can sort of reproduce the data firstly in a different cohort, and see if we can get some further understanding in these particular subsets.
Yeah, it's kind of a new frontier because you have these new tools and these new,
Yeah.
samples and your new approach. I mean, for a while it seems as if the B cells were kind of in a corner, left in the corner. But I mean, together with your work and others, they're really coming out into the forefront. It's fantastic.
Yeah, Mia Smith's done some really nice recent work on anti-insulin B cells, and sort of almost finding some similar phenotypes in terms of the CD11C, that seems to be sort of enriched in these anti-insulin B cells, and so she's been doing some really nice work as well.
She has. She just gave a presentation to the working group last month. And we'd love to invite you to join us. Just you want to stop in and check it out, see if you have any interest in being a part of it. I'll send you a PM. But
Thank you. I love that.
yeah, it does also. I guess the last question I had for you is, what are your thoughts about, kind of as you develop um, these markers and using this in, sort of during the prodrome as biomarkers. Does you think it even has a possibility of B cells, indicating progression in some? I
Yeah,
mean, it's
so
a huge question, a big ask,
yeah,
so.
yeah. So I was hoping that I'd be able to. I've got a close collaboration with Kathleen Gillespie and the Box study at Bristol,
Mhm.
and she's a collaborator on the most recent grant, to see if we can get some donors who are multiple autoantibody, and see if these multiple autoantibody donors will subset as well, and whether that's something that would be able to pull out, if it's, these DN2 B cells are associated with a loss of, rapid loss of C-peptide, and whether we could see that in donors that haven't reached the diagnosis yet, would be really interesting to see if that we'd be able to detect them in the earlier stages.
Yeah, further teasing out
Um,
the endotypes. That would
yeah,
be fantastic.
yeah. So lots of work to do.
Yeah, you do have a lot of work, but you have a
Yeah,
lot of great contacts and collaborative efforts. As you mentioned, several of the other women scientists who are going after this. It's a really exciting time in the B cell world of
It
T1D.
is.
So thank you again, Joanne. We'll keep an eye on it. I'll send you a message after the talk. Thanks again for talking with us and sharing your work.
Oh, thank you so much. Bye, everybody.
Have a great rest of your day. Bye-bye.
You too. Thank you. Bye then.